Hi there, and welcome to my presentation. My name is Gregg. I'm a recent graduate of Northwestern Connecticut Community College, and I'm also a participant in the Howard Hughes Medical Institute C-Phages program. So, to begin, we know modern medicine has come a long way in a short amount of time. But as time progresses, new and more challenging problems emerge, and the medical field is by no means unaffected. One of these problems is the rise in antibiotic-resistant bacterial infections. This issue has not only left hospitals hopeless to save some patients, but this issue places healthy individuals at risk. If this dilemma continues, going to the dentist to remove a tooth, even having an embedded splinter, can place individuals in potentially life-threatening situations. This has led scientists and researchers to search for alternative treatments, reviving an unusual treatment discovered before the creation of antibiotics: bacteriophage therapy. So, considering the damage viruses are capable of, it seems counterproductive to treat a bacterial infection with a virus. Bacteriophages, however, are a virus that specifically target bacteria and not human cells. Bacteriophage are found everywhere on Earth, including the gut biome of humans, and in great numbers and types. It's their selectivity, abundance, and diversity that make them so valuable to study as an alternative treatment for curing the most virulent superbugs. So now that we have a little background information about the project, let's take a look at the poster. The ability to contribute to bacteriophage research was made possible by the Howard Hughes Medical Institute, a nonprofit research organization located in Maryland. The C-Phage project is a student-sourcing undergraduate research program founded by the Graham Hatfield's group at the University of Pittsburgh and the Howard Hughes Medical Institute Science Education Division. Our purpose was to discover new bacteriophage through established microbiology techniques and characterize them through the genetic annotation and bioinformatic analysis of their genome. Among the 37 genes studied, we could only validate 5 promoter regions. They were found upstream of genes 38, 45, 48, 63, and 71. Conserved repeats were located by genes 28, 33, 34, between genes 66 and 67, 68, and 69. The significance of repeats in genes 28 and 34 were undetermined, but 2 repeats located within gene 33 code for topoisomerase primase, an enzyme that assists in the removal of supercoiling in DNA. Much is still unknown concerning phage research. Each annotation project, however, brings us closer to discovering life-changing technology and medical breakthroughs. Through all the hard work carried out earlier this year, I feel it's worth noting that our efforts were fruitful. Gordoniaphage GemG's genome was fully annotated this spring and submitted to GenBank for review the following July. Our work was recently published on September 29th of this year. Despite our success, however, there were some minor limitations that are worth mentioning. To begin, DNA Master called 91 promoter regions in total between base pairs 23,001 and 46,185. Between DNA Master and the results from SoftBerry's BPROM program, only 5 promoter regions could be verified. DNA Master has a genome sequencing accuracy of 80%. So, with this in mind, it's possible that BPROM sifted through this 20% error margin and found what was accurately generated by DNA Master. Another potential limitation of this project involved the range and education levels of its participants. However, we clearly stated our goals and provided introductory material to the high school students at the beginning of this project, explaining the biology behind bacteriophage and how we will be studying them. Well, it looks like we've reached the end. Thank you so much for taking an interest in this presentation. This project has led me to develop both a strong fascination and admiration for these microorganisms, especially after learning about the numerous benefits they have to offer. And as a future researcher, this also served as a great opportunity to gain experience and to learn more about the field of research. Bacteriophage could be used to help those allergic to antibiotics or those with compromised immune systems, and even go beyond the realm of medicine to serve in dilemmas surrounding the environment, agriculture, the food industry. Truly, the list goes on. It may even give us insight on virophage therapy to use as potential treatment for viral diseases. I look forward to answering any questions or addressing any comments you have about the presentation. So until we meet in November, take care and talk to you soon.